Say you found a skeleton. How would you calculate that distance? You personally, how would you decide?
Well, interesting question. I don't know. I'd have to do some research on that.
Hello, everybody, and welcome to the after show. I'm the Dapper Dino. Nessie20 is, I think, off getting water or something. Not really sure. But
no, I'm back.
Oh, there he is. Before we get into anything, we do have a subscriber chat from Carrie Bronson, who's been a member. Sorry, a member chat from Carrie Bronson, who's been a member for 7 months at the level of Amniote. And he says, "I invoke the blessing of the void upon all here. May its eternal vastness consume your troubles, and may its absolute tranquility bring you peace." Thank you very much. And of course, for those of you who don't know, I, the Dapper Dino, do sort of run the Church of the Void, which encourages you to give up your troubles to the Void, which will consume them and dissipate them. And also, the Void accepts donations. So I'm going to start off by just asking Nessie20, what did you think of the debate?
Well, I think it went well, like in terms of no interrupt, or not actually no interrupt. I think, if I remember correctly, it was really well moderated, and everyone got to say the piece, etc. Although I would like one of the main issues I have is that the topic was very vague. Like, of course, we had the topic of novelty and evolution, but he went from junk DNA to the Cambrian explosion, yada yada yada. So it was not really focused to my taste.
I agree that it went less focused than I would have liked.
Yeah.
Unfortunately, as the moderator, I have to tread a pretty fine line between, saying, "Hey guys, you have to stay on whatever the topic is," and being neutral enough that me saying that isn't going to favor one side over the other. And since both sides were willing to engage with the various things that were brought up, and they weren't completely unrelated, like it wasn't Mr. Batman asking me how I know that, abusing children is wrong, that I kind of allowed that. And if Dan were here, I would tell him I wish he had ignored some of the things that I think were rabbit trails. But
Right.
I understand why he didn't. And so, I do agree that it went off. If I had been on Dan's side, my point probably would have been like, hey, look, here are novel functions and novel traits that we see evolving in real time right now in human history, observable by humans.
Yeah.
So whatever else you bring up, whether you bring up the Cambrian explosion, how could the trilobite eye be the first eye in the fossil record, any of that doesn't matter.
Yeah,
we have known examples of evolution creating novelty. Either refute those or I win. That's what I probably would have gone with.
Yeah, I would say this debate is not on the Cambrian explosion specifically. We are just arguing the question of evolutionary novelty, and I'm showing you examples, so let's address them.
Right, because I think it went a large portion, I think, of Finding Truth's argument ended up being, if you can't explain every single example that I pull out, therefore evolution can't in any circumstance create novel function, which is logically fallacious. That's not how this works. Now,
so basically, the topic went from can evolution explain other evolutionary novelties to can evolution explain all novelties, like every single one. Yeah.
So I would, if Dan shows up, and he might, I would have pointers for him. Oh, and here is the man himself, Ahmed. You are in the chat, Ahmed. How are you?
Hi, how are you? Hi, Nestle. How are you doing?
Hello. I'm having a deja vu moment.
Oh yeah,
we're all together again, the three of us.
yeah. It's always fun to be here. So, are we starting now or in half an hour? I'm a little bit confused.
Oh no, we were already started. We've been live for about 5 minutes.
Oh, okay, all right.
Yeah, no worries.
Hello.
So, I will do what is traditional for me, and I will ask you what your thoughts are on how the debate went, and what you thought maybe you could have done better, what Dan could have done better, anything like that.
All right. I think it was nice in terms of being calm. I like this kind of disposition.
Mhm.
I think the topic was pretty big.
Yeah.
So I wanted to cover all the points that he raised, but I think it would have taken 4 or 5 hours or something. So I think it calls for splitting it up. There was so many topics there. Things about endosymbiosis, about eukaryotes, about simple mutations. So there was so much stuff. So I think even there were questions that we couldn't have the time to answer. So maybe some of the audience will be here. Yeah, maybe some of the audience will be here. I think Dan, on his part, I think he. I think I did very well in the debate in terms of responding to his topics. On his side, I think he might have done better. I think
by doing what?
Maybe by presenting fewer cases with more depth.
Okay. Well, I think that ties back into the. It's a fairly broad topic. It's not as broad as some of the topics I've seen in some of these debates. I've had people say, "Oh, why don't we just debate creation versus evolution?" I'm like, nope, that could be almost anything. That's way too much. So before you got here, I was expressing that if Dan were here and I were to give him pointers, I would say that I think a few too many times he allowed the conversation to go away from the core topic. Now, I do think to some degree that's because you did bring up things that I don't think really address the core topic in the way that I think would have been most useful for the debate. But he also allowed that he ran with it. So, for instance, I'll just go ahead and tell you what I would have done in Dan's place, because I think, as we all know, that while I was neutral during the debate, I do in fact tend to agree with Dan on matters of science more often than finding truth here. I don't think that's a surprise to anyone. But so what I would have gone with is, so if you remember, in his opening statement, Dan brought up several examples of new traits evolving during human history that humans were able to observe happening in real time. And then I would simply say, if this happens, and we know it does, therefore new novel traits can and do evolve, case closed. What would be your comeback against that argument?
Well, yeah, my comeback will be it's not. I don't have an issue that novel traits have evolved. Well, because obviously I have myself given examples. I have said in one of the comments in one of the rounds that, for example, studying human genome reveals more than 1,700 novel genes that do not have homologs. So it is common to find novelty. Now, this is not the question. The question is whether this novelty is driven by natural selection acting upon random mutations or not. Whether the mechanism.
It's the question. The question is. Oh.
Okay, yeah. So I actually do have a question though, because I'm currently looking at what I have for the debate, and I'm looking at the thumbnail where I announced the debate resolution, right? So the resolution for this debate is evolution can create novel features, with Dan in the affirmative and you in the negative. Is that not what we agreed to?
Yeah.
Okay. Well, you did just recently say that novel traits do evolve. So are you now conceding that the answer to that question is yes?
No, I'm saying that novel traits appear.
Okay.
Now, if you are using the word evolve in terms of evolutionary theory mechanisms are responsible for them, of course, my answer will be negative. Okay. There are novel traits all over the place, since Luca until human beings. The question is, are they produced by what the theory claims to be the mechanisms or not? My answer is not.
Okay, so let's take a concrete example, right? So we have, and of course we're going with the modern theory of evolution as it's understood in professional biology now. We're not worried about Darwinism or neo-Darwinism or anything else that has been since expanded upon up until, now in 2021. So let's say that we take the one of the novel traits that Dan mentioned, say quote-unquote placentas in lizards. Now, I say quote-unquote because they're not identical to the placentas that we have in mammals, but they do perform the same function, and they are novel traits among lizards. Is that... Where is that coming from? Is that coming directly from God, and how do you know?
I think we have something related to citrate on the screen, so I'm not sure what...
We'll address that one after the lizard.
Sorry. Yeah. Let's go for the dapper first set. So, sorry for showing this.
It's okay.
Yeah.
So, is it your contention that the genetic pathways required for certain lizards to use a placental type development for their young, is that coming directly from God? Or are there mutations? Because I'm going to tell you, before we get into this, we can trace the mutations. So if you're going to say it's coming from God, I'm going to want a really, really tight argument as to why that has to be the case.
Okay, so the topic of the debate, we had a debate before about mutations specifically. I think it was me and you. But in this one, we're talking about novelty. So when I'm considering novelty, and you are putting, for example, placental mammals or whatever, placental reptiles or whatever, use of placenta on the screen. Yeah. So I will then ask, regardless where you trace those systems, the placental system to, the question is, when this system became novel, how did it come? Not how did it change after it arrived.
I agree. So that's the question that we're asking.
Yeah, that is exactly the question of the debate. When you are looking at novelty, the topic of the debate, it would be the arrival of the placental system to the stage. Even if you are looking at it in lizards or in reptiles, whatever, I don't mind. But then you will say, okay, we have some lizards using placentas today. This can be traced, I don't know, back a few million years, where this had this kind of ancestry. I don't mind. And then the question will be, how did placentas arrive to reptiles in the first place?
Right.
This will be the question.
Is it your contention that happened because God directly put those genetic changes into some reptile way back? Is that your contention right now?
Okay, so I would not say it is my contention. It is my worldview.
So you are currently advocating for that as the case, right?
Yes, I'm advocating that whatever novelty that is there in the whole, I don't know, in the whole tree of life, if you would like to call it,
okay,
or in the whole compendium of organisms, whenever there is novelty, it cannot be attributed to the modern synthesis or the extended model synthesis mechanisms.
Okay.
Because all of them, essentially, at the end of the day, the core of them, at the end of the day, is chance or pure physical properties.
Okay.
Pure physical properties plus chance cannot produce novelty. This is my position.
Okay, so
there has to be a mind behind it. You can call it God or whatever.
Okay. Do you agree that the differences which allow some lizards to have placental development is genetic? That is the foundation of the difference. Genetic.
I would expect so.
Okay. So are there some genetic changes that cannot occur because of mutations?
Can you explain the question further?
Yeah, is there some change that could happen to a genome, right? Like, let's say,
yeah.
I don't know, do you have children?
Yeah.
Is there any particular genetic change that you could find if you were to do a complete genome sequence of your children that could not be explained by a mutation?
Well, if you define mutation just as change, then your question will be: is there change that cannot be explained by something other than change? And the answer will be: you have just stated the word in a different synonym.
Let me put mutation in terms of the context of a mutation that was not, or change that was not directed by an intelligent agent.
Okay, that's different. So that's why I always insist to put the qualifier random before the word mutation. So if you're asking me if there are some changes that cannot be attributed to random mutation, then yes, of course, the arrival of the human being altogether on stage cannot be attributed to random mutation.
How do we tell which genetic changes cannot be the result of mutations without special pleading?
Okay, so if you look at human beings who exist today, and you find a change to somebody's genome, to Ahmed or to Dapper or to Neslik, I don't mind that this is a change that has resulted due to random mutation.
Okay.
But if Ahmed grows wings tomorrow, okay, or becomes an X-Man and is moving things or causing storms or whatever, like the science fiction thing, then okay, I will definitely not accept that this is the result of a random mutation.
All right. So we have right now, we've got the two big kinds of changes that we couldn't reasonably attribute to simple mutations are borrowing whole sections of genetic pathways from other organisms that aren't in our line of descent, which would be sprouting wings, and two things which would otherwise simply be miraculous or magical anyway, which would be gaining powers that can't be normally explained by the laws of physics. Which of those happened going from exclusively egg-laying reptiles to having reptiles that sometimes use placentas or even habitually use placentas and sometimes use eggs? Which of those happened? Was it genetics that was borrowed from another lineage, or is a placenta magic?
Okay, we can keep with the human, and we can then refer to the.
I would prefer not to, because that is switching the topic.
Yeah, because we were giving an example with humans.
Well, you gave an example with humans. I would like to stick to the example I started with the lizards.
Yeah. So if you find at the end of the day that you are tracing this feature, I did not study this matter of placentas in reptiles, to be honest with you. But let's talk genetic.
Well, let's just make a prediction. Do you think
which
do you think? Do you think placentas are magical and they can't be explained by physics, or do you think that these genes had to actually be borrowed from a distant organism where there's no common descent that leads to those genes?
Okay, so my argument, just a...
Oh, I mean, Nestle, am I asking the right question there?
It's really...
Yeah,
he's back. Never mind.
Yeah. So the thing is, I just want to be very clear here. You take an organ or an organism, which is a reptile, and a specific system, which is developing a placenta connected to its whole, reproduction system, okay, as part of its reproduction system, okay? And then you want to ask, a reptile is now giving birth through a placenta. Is this novelty? You will trace it. If it is there as a capability of its ancestor, just reach the rim of where it is not there anymore, where it is really novelty.
Okay.
And then if you come and ask me, what is your expectation? My expectation will be that this thing cannot be explained by random mutation.
Okay. And when I asked you how to know which mutation or which changes to genetic code couldn't be answered, your answer came down to either you're acquiring traits from outside lineage to which you don't have direct ancestry, or it's magic. Which of those two is the placenta in your estimation? Which one would you project? Are placentas magical, or are lizard placentas actually borrowed from another lineage, like maybe mammals or something?
Okay, so the option of magic does not exist. I don't know why you consider that this is an option that I have proposed.
Well,
So
because
this is
you
not
proposed that one of the options for it being obviously not natural, unguided mutation would be if I develop powers like the X-Men. I would consider all those powers to essentially be magical because they all violate the laws of physics.
Yeah.
Okay, this was more like a rhetorical example, but
what
I meant is something that is not typical that humans can do. Okay, so I don't know if humans, for example, can sense things electromagnetically like sharks do, or things that are known not to be there in the human genome suddenly become part of what humans can do.
Actually, humans do have weak
Did- did-
electrosensitivity.
And also,
I'm sorry?
didn't that exactly happen with like lactose tolerance? Like there used to be a time when nobody was lactose tolerant as adults, and now some are. Right.
Yeah, that's why I used an example that nobody will say that sometime another creature had it, or it might be available in the human lineage. So I used a fun example, but I mean the concept is obvious. Yeah.
So
if I'm back to your question,
yeah, go ahead.
if we go back to the lizard thing, and we go to according to the theory, to what is an ancestor on the lizard lineage that is known not to have, or the analysis shows that it will not have any of the I don't know, the evo-devo mechanism and the genes that will eventually end up having a placenta. And then you ask me, how did the placenta come into existence? I will say that the designer of living things caused it to be there. There is nothing random about it. This will be my contention. The evolutionary paradigm will propose that, I don't know, gene duplications took place, and then random mutations, and then neutral theory, and then whatever, and then promoters happen to occur at that place, and then a start codon and an end codon happen to be there, and then some change, maybe it comes abrupt, and we'll call it I don't know, punctuated equilibrium, whatever. We'll throw terms around it, and things that are completely impossible to occur to me by chance, and then and that's how it came about.
Okay.
My thing will be, maybe yes, at the end of the day, some genes will have to reflect the function, because we are in a world that is governed by laws of physics and chemistry at the end of the day. But those genes did not happen to develop this function because the DNA mutated randomly. No, because the DNA changed because an intelligent agent caused it to change.
Okay,
no
so
random.
you listed a whole bunch of things that could happen, right? Which have been proposed as explanations for novel genes and novel features. So you mentioned gene duplication. These are very simple questions, yes or no. Does gene duplication happen randomly, or does it always require an intelligent agent?
I don't know if even somebody in the evolutionary side can make an assertion that gene duplication is purely random. I don't think that somebody can do that.
Well, ultimately, nothing is purely random. It's just that we don't usually have the ability to predict it. So, either way, do you think...
Well, it's maybe better to say they are not directed to a specific outcome, perhaps.
So does
it's like poker. It's like poker. Like the types of cards you get, or combination of cards you get, is not entirely random.
Right.
But it's still not like right. It's not the directed like for a purpose or something.
Okay, so this is yes or no. Can any gene duplication ever happen without it being directed by an intelligence?
I would expect so. It will.
Okay.
Yeah.
Can a start codon ever be inserted or have a point mutation to create a start codon anywhere in the genome without it being directly directed by an intelligent agent?
Yes, we know that random mutation. We know that mutations take place, and there is a chance, maybe a small one, that a mutation causes exactly one of the sequences that will be recognized by the transcription system as a start codon. Right. Yeah.
Okay.
It's possible.
Can a promoter region be inserted into a genome either by point mutations or by transposition or by duplication or any other means without it being directly imposed by an intelligent agent?
Yes, it can.
Okay.
Yeah.
So And point mutations in general can happen without it being directly caused by an intelligent agent. They can happen unguided.
They can and they do.
So what you're telling me is, you just said that this sequence of things that, quote unquote evolutionists give as their plausible evolutionary pathway that you say is impossible is a sequence of things that are all possible.
Yes. So it is just like that. Is it possible that you throw up to the air a bunch of 200 or 300 playing cards and they fall down in the form of a house of cards? It is imaginable to happen, but is it possible to happen in the real world? The answer is no.
Okay, so the argument is just that it's extremely unlikely.
In terms of the genome, it's actually very common. Like I remember a paper where it says that in the genome of E. coli, it's actually under selective pressure to prevent accidental promoters. So accidental promoters by pure random mutation is very likely to occur, but they're actually under selection to prevent accidental promoters.
Yeah, and here's another thing I would point out. While I agree that, let's say we went to, an electronics store and we bought a bunch of, wires and resistors and capacitors and whatnot, and we threw them on a table, it's unlikely that they're going to form a circuit that does anything. However, when we experimentally put together random, peptide chains, they do things routinely. So it's a disanalogy because we know for a fact that when you just toss random peptides together, you get function. Whereas we know that generally when you toss electronics on a table or electrical components on a table, you tend not to. So it's not a very good comparison because we know it happens in one case and we know it tends not to in the other case. So we're talking about the case where we know that just random assemblages of nucleotides, essentially, because these peptide chains are programmed by nucleotides in nature. So a random chain making up a random protein is functionally the same as having a random set of nucleotides being transcribed into a protein. So we know that can produce function. So it is absolutely 100% known to the highest degree it could ever be known that, yes, random DNA sequences can make functional protein without any direction.
Do you want me to respond to that?
Yeah.
Okay, sure. So let me tell you this. I'll use the same analogy that I used in the discussion that you have used now. So let's say we have this test bed where we are throwing electronic components on them. Like we have this test, polymerization machine that just polymerizes nucleotides or polymerizes proteins. Now, when you throw an electronic component onto this test bed, a resistor will deliver the function of a resistor. It will resist electric current. A capacitor will deliver the function of a capacitor. It will resist. It will cause capacitance. It will accumulate charges. Now, if you throw plenty of them, okay, then this is the question: whether they will come together. And I'm talking about a test bed, a test table, where you have those perforations, and you will just plug components without any order. This is what you are saying. We are randomly polymerizing nucleotides or peptides. So at the end of the day, if I am telling you, if somebody comes and tells me, I will just put on a test bed, on an electronics test bed table, a bunch of 100 components, and I will do it so many times, and I will wish at one time that one of those configurations will be a device that have function, I think this person cannot really be reliably employed by any electronics design firm. Now, yeah. Let's now. Yeah. Yeah.
Right. It's context dependent. Yeah. Right. Yeah. I actually have seen a paper where they say that like random proteins often bind to metals, and metal binding is a very, very important function in our metabolism as well.
Okay, so let's take it to the other side on the biochemistry side. So let's now look at your example. You want to polymerize a set of amino acids and produce a peptide chain, produce a protein. So at the end of the day, now why this doesn't work, in my opinion, as an analogy, because this is not what happens in living systems. You just don't go and polymerize peptides into proteins. You need to create code for a protein. It needs to be assorted in a specific way. It needs to have a start and an end. It needs to have a promoter, and it needs to have a transcription system. And the transcription system will then check against the rules. And if this is not a gene, according to its perspective, nothing will happen. And then if this is a gene in its perspective and it produces a protein, a function is contextual. If it produces a protein, it will be some more protein debris that is thrown in the cell if it does not go into direct harmony with the systems of the cell. Apart from that, if the cell sees that this is complete gibberish, what you can call, you can give it a nickname, the DNA immune system will just kill it. It will mute it. So we are not looking at systems that typically produce things from random stuff, because just reading the random stuff needs the rest of the system.
Okay,
so,
but Ahmed, your point of the reason you brought up this analogy, right, was because you wanted to say that random mutations in a genome couldn't create novel function. Now you're changing that to, you're saying that random assemblages of nucleotides couldn't create a genome. You realize that those are different arguments, right?
No, I'm just now saying, responding to the example that you have given, because you have started from polymerizing polypeptides directly. Yep. But what I am saying is actually even directly what I said in the debate. If somebody comes and puts a proposal, which is essentially what the evolutionary paradigm does, that in an intergenic region, okay, mutations will cause start and end codons and a promoter, and what is in between will be accepted by the system as delivering novel function. Assist an organ, a new major thing, not just a change to an existing thing that used to work, then I have very big problems with that. This is the definition of novelty, and novelty cannot happen. If it is possible that it happens, and this is what has driven life, where are the real life examples where really complete systems, like placental systems, for example, that you have mentioned,
I object to part of your definition for novelty, which is that it requires an entirely new organ that isn't a modification. Evolution doesn't really propose that things occur that are not modifications of previous things. So, for instance,
even the novo genes are from non-coding genes before, so that they are like starting with something.
Strictly speaking, if you're asking for a new gene to simply pop out of nowhere and enter into a genome from the ether, you are asking for evolution to do something that it does not predict.
Yeah, it's also that you're asking that question to evolution, not creationism, because creationism is actually making that claim and not evolution.
Further, that point also doesn't counter the idea that even if you were right about that prediction of evolution, it doesn't counter the idea that mutations, which are unguided, can and do produce novel traits, novel biochemical functions, and eventually could lead to the evolution of new organs, which is already happening, as we've said, in some species of lizards, which are getting more and more towards the placental end of development. And again, with the caveats that the placenta, as we think of it as a true placenta in mammals, is not the same structure that you're getting in these lizards. That's, I don't want to have to say like pseudo placenta every single time. Sorry, guys. So there's a big push to do a couple things from what I'm seeing. One is to retreat from current observable instances of novelty to the relatively hard to observe past and asking how novelty occurred at that point without addressing the current novelty that we're seeing arise in modern organisms. Another one that I'm seeing is an attempt to rush into a bad analogy to dismiss the possibility of unguided mutations being the source for these novel functions. And also a willingness to equivocate as to why that analogy is supposed to get rid of this. And this is not so much a thing I'm observing as sort of a point I would like to make about analogies. Analogies are handy to help people understand technical things that are not familiar to them. They are not good at being an argument unto themselves as to why something in science is or is not the case. You really have to go into the actual event as it's happening. So if your argument for why unguided mutations can't create novelty involves an analogy, you've already kind of failed before you started the argument.
Okay, so that's why in my short presentation, I'm not sure if it was short, but it's definitely within the time.
Absolutely, which is what mattered.
Yeah, I did not give analogies. I just referred to the comments of Muller himself in 2016, where he has indicated 3 specific categories: phenotypic complexity, like eyes, ears, and body plans and anatomical structures. That's why I am referring to organs and systems of organs. Phenotypic novelty, like what we see in the Cambrian explosion, for example, and the mammalian radiation. And the abrupt discontinuities in the fossil record, which will essentially, at the end of the day, maybe boil down into the first 2. So I'm not looking at maybe something like lactose intolerance or I don't know antifreeze protein. We're looking at these things that essentially at the end of the day constitute what we will see as a different creature, as a different function, as a different arrival to the stage. Bats that can use, I don't know, supersonic or ultrasound vision, sharks that have something like a radar, the arrival of mammals altogether, the arrival of multicellularity, the big events, the real thing, arrival of vision, of hearing, of tastes, of digestion, etc. These are the things. Without these things, there are no creatures altogether. We would have stayed in a creature like Luca. For the whole 4 billion years.
Okay, but
how did that happen? This is the question.
But do you
not how did some existing feature get modified, maybe epigenetically or genetically or whatever? This is a different topic.
Okay, but do you see why, like you said, that is a different topic than the one that you agreed to debate?
No, I agreed to debate novelty. I can also agree to debate mutations.
So here's
the thing about novelty.
Let me get this out here. Here's the thing about that debate topic, right? What it takes to confirm the "can evolution create novelty" statement is any example from any time in history of evolution doing the thing. Once you have one, the answer is yes. If you want to say, but I think it probably couldn't have created this particular novel function that I'm going to point to, that's great. That's a separate debate. The debate you agreed to, and I think both you and Dan went away from this topic, so I'm not saying that this is, an all on you. I think that had I been involved in the debate in a less neutral capacity, I may have wanted to insist on bringing this to a more pointed discussion. So the debate topic, can evolution result in novel function? And we're seeing it now. We're seeing it with lizards. We're seeing it with many animals that are gaining things that are analogous to chloroplasts. We're watching it happen with, we saw it happen historically with HIV coming from SIV, where it now gained the ability to antagonize human tetherin, which is a protein in the immune system of humans, which prevents most SIVs from attaching to cells in human hosts. So all of these things are happening historically. Is it your contention that each one is the direct result of intelligent intervention in the genomes of these organisms? Because if it's not, then the answer to the debate on both sides is yes.
So my contention is that those examples that you have given will fall under three categories. One of them is that they are not an accurate representation of the fact. And another one would be that they do not classify as novelty, and I can explain why. And the third one would be that I do not accept that they can be a result of the evolutionary mechanisms, and I will require evidence.
Okay.
Yeah. So if, for example, when you have mentioned that some animals are gaining photosynthesis, the examples that I know about is that some animals are piggybacking some algae with them, and that there is an endosymbiotic relationship. Not that the animals are becoming photosynthetic.
What is the difference between those two things?
These are the examples that I know about. I have not read a paper. If there is, send me, and then I can critique it if I take enough time to learn it.
Well, I believe Dan is listening, and he does have those papers in his library of sources. So Dan, if you could forward those to Finding Truth, because I do suspect strongly that you're listening, mostly because you said that you were listening. So I'm assuming that you weren't lying about that. So
yeah,
there's a few things. What is novelty to you then? Because the only one that I've heard that isn't either accusing scientists broadly of dishonesty, and that isn't the only one really, that I heard there, or that isn't just sort of meaningless because saying like, oh, I don't think it happened because of evolutionary mechanisms. Well, that's just restating your position. So the only other one there is you don't think it's actually novelty. So what is novelty to you?
You said something very important that I have to comment on. So when a lizard or I don't know, a reptile is carrying some algae with it, and this algae is in an endosymbiotic relationship or a symbiotic relationship with it, so the algae is using photosynthesis to provide some benefit to the animal. Representing this as the animal has become photosynthetic is not an accurate description of the fact. This is what I'm saying. So it cannot be used as proof for anything other than symbiosis.
Okay, but can you answer the question?
This is the first category, okay? So answering your question, does this category count as novelty? No, it doesn't.
Not the question. That's not the question.
Yeah.
The question is, could you define novelty so that we can know which things you would and would not accept as novel, so that we don't talk past each other? Because giving examples and saying, I don't think it counts, doesn't tell us what your definition is. So when you're done with your definition, we should all here and in the audience be able to look at any trait that comes up in an organism and tell whether or not it is novel without needing to ask you about it, assuming that we've sufficiently understood you.
Absolutely. So that's what I did in my presentation. So they fell into two primary categories and one additional one that relates to fossils. So the first one is phenotypic complexity, and this is from the Royal Society meeting, from an authority like Muller, not from me. And this is defined as the origins, for example, of eyes, ears, body plans, and in general, anatomical and structural features of living creatures. The second one is phenotypic novelty.
I think Muller has published a paper on like a particular novelty, the muscle of that was the tibiotarsus of birds, if I'm not mistaken. Of tibio, yeah.
That actually would be a good question. So you said that various kinds of anatomical novelty might count. So would going from having 3 separate tarsal bones to having a tibiotarsus, would that in fact be an example of novelty?
So the examples that I have given, for example, are eyes, which is a system for vision, a system for hearing, a new body plan. But if you are telling me that a bone structure is different, or this one has 1 bone, this one has 2 bones, I will not count it under this discussion.
Okay. Well, then I'm gonna create a new rule for definitions for this live stream. I'm gonna create a new rule for definitions for this live stream. You are not allowed to give examples until you are completely done with the definition part. So, starting again, what is your definition without any references to examples until after you're done with the definition? for novelty?
Okay, so let's take the phenotypic complexity. I'm a bit getting out of my league here by trying to put a general definition for something that needs examples. But I will say systems. I will just summarize it into, because systems involve complexity. Okay. So eyes, ears, body plans, whatever will fall under systems. So I will not then, if you tell me a bone or becomes two bones or fingers become merged into a bigger hoof or whatever, I will tell you, no, I am looking for the origin of chordal systems, of skeletal structures, of exoskeleton, of vision in general, of eyes, which will be vision will be eye and a neural link and a brain that understands it. Listening would be the ear and the sensing system of sound and the neural system, etc.
So
systems.
At what point does a trait go from not being a system to being part of a system or being a system? How do we know when something's a system and when it isn't?
The definition of what counts as a system is, I think, quite refined in biology and anatomy. So I don't need to define it.
Well, you do though, because you've used an undefined word in your definition of complex or of novelty.
So you will call the digestive system is a digestive system.
Nope, that's an example. Yeah. We can't use examples until after you've defined it. So first define it, then you can give some examples.
So a system would be a collection of organs and parts that will deliver a specific function. In this case, since we are in the context of novelty, the function needs to be not existent before the system exists.
Well, we're just asking about this. So in order to count as novelty, a new collection of organs has to form. Do they have to form all at once? Can they form one after the other?
So that we call it a system.
Well, do they have to form all at once?
I'm sorry, the question again?
Do they have to form all at once for it to be novelty?
They don't have to form all at once, as long as what you are saying is that there are miniature versions of the system. Is that what you are saying?
Well, let's go with this. Okay, so let's say I have an animal that doesn't have blood vessels or a heart, but it has some fluid inside that sloshes around and just kind of mixes up things like oxygen and nutrients, which, by the way, we have many animals like that. Is that a circulatory system?
Most animals have that, actually.
Yeah.
Yes.
Is that a circulatory system?
No, it's not a circulatory system.
Okay. Now, we have some animals that have a thing like that, where they just have a fluid that sloshes around. But somewhere in their body, they have a single muscle that contracts and releases that gives it a little bit of flow. So this is sort of like a very basic one muscle, single chamber heart. Is that a circulatory system?
No.
Okay. So now we have some organisms with that open circulation of this fluid, right? But they also, and a single little muscle that pumps, but they also have little areas that they have as little indents in their body that take in extra surface area, which allows them to have extra oxygen going in. Is that a circulatory system?
No. Okay.
Now we have some creatures where instead of just one little muscle, they have a chain of them that all pump in sequence to give a stronger flow direction. Is that a circulatory system?
No.
Okay. Now we have some where they expand how much of this system is enclosed in some kind of membrane. Not always musculature, but this gives increased directionality and they can direct some amount of this fluid more effectively to different parts of their body. Is that a circulatory system?
I need an example to understand what you're talking about.
So, for example, spiders can actually direct this fluid into their legs, which is how they extend their legs. And so they do require additional channels through which it's called hemolymph, but it's sort of like blood. And so they use the pumping of their hearts and these channels that they can pump through in order to extend their limbs. Is that a circulatory system?
If you are talking about a creature that has a heart and it has a fluid that delivers what a circulatory system does, then yes, it is a circulatory system for insects, at least, or for this kind of insect.
In that case, one of the things that you said a while back wasn't a circulatory system is one, which is just an open amount of fluid with a single muscle in it somewhere that helps give some extra circulation. So that is a circulatory system.
No, because you are now talking about a system that has a heart that is closed loop. If you are describing something that is a closed loop, If you are not, then I would say no. But if you are if you have something that has a heart and has blood, and the heart pumps blood around.
Okay, so it is the case then that an open circulatory system, like we described earlier, with a single muscle that pumps and moves this fluid around a little bit more effectively, is a circulatory system.
Yeah. So if you ask me, does the muscle in the leg, I don't remember the name of the muscle, which if you are exercising and if you have good muscles in your legs, it will help pump your blood up. Is this part of the circulatory system? No, it's not. It helps push blood maybe, but the circulatory system has a certain definition. So we cannot play with the definition.
Okay, but what I'm pointing out here is that we started with something that essentially didn't involve any organs, really. It just involved a bit of fluid sloshing around. And then with the addition of one muscle, we got to a system. And one muscle isn't even technically an organ. So I think your definition of system is lacking, because if what you're telling me is true, that, say, a single heart muscle and an open circulatory system, an open circulation, right, is a circulatory system, you've really only got one. You don't even have one organ there. You've got one structure that can barely be called an organ, because traditionally organs have to involve more than one cell type. So
I'm not an expert on spider anatomy. I don't know if the spider system is
we're
open.
not at spiders yet. We're still like way back in like very basic bilaterians.
I will. Okay. So you're back to the initial examples. No, I will not count that as a circulatory system. No, I will not count.
Okay. So do flies have circulatory systems?
I will call it something else that relates to the context of that creature.
So
what should I call it? I don't know.
I think they have what's called a
creature has fluid
right?
in it. So good enough. The creature has a fluid inside it. So I'll give you. Let me tell you something that's more obvious to me. So you have those fluffy putative creatures that were there in the Ediacaran, and they, I don't know, absorbed nutrients from the ocean or whatever. Is this a digestive system? It's not a digestive system.
I don't know. That's the thing. I don't think there's actually a very good definition of system.
Because then there is an assertion that digestive systems, digestive tracts, came novel to the stage in the Cambrian explosion, where you have food come ingested.
It's
probably not. It's probably before
No. Actually, it's certainly before the Cambrian explosion.
Yeah.
It is absolutely not the case that internal digestive tracts came at the Cambrian explosion. It was before that by a lot.
The last time I checked, I found that it is in the Cambrian, but this is even a technical issue. We have
Ediacaran animals that are of various groups, including almost certainly cnidarians, stem mollusks, stem arthropods. We have even deep into the Precambrian, we have stem bilaterians or possibly crown bilaterians that are extremely basal. And all of these groups have internal digestive tracts. Some of them are blind, so they're not exactly like yours. But yeah, so we know for a fact that internal digestive systems predate the Cambrian by a lot.
Good, so fine. They predate the Cambrian or not. I mean, the example, the comparison that I'm making between something that absorbs or adsorbs nutrients from an ocean, it serves the same purpose. It gets nutrients in, but it's not called a digestive system. So right. if you will extend the definition of a digestive system to anything that will bring nutrients in, this is an overstretching just to serve the purpose of the argument.
But the thing is,
it proves nothing.
All I'm pointing out is that we don't have a good definition for system that we can apply in an objective manner across diverse taxa. Therefore, since your definition of novelty relies on the word system, and we don't have a good definition for system, as I think this entire conversation has demonstrated, your definition for novelty is inadequate because it relies on an ill-defined term.
I think then the guys who have met and said that the appearance of novelty is not properly justified by the mutative mechanisms, and that we need to add the following 5 or 6 categories into extended modern synthesis had no case. Why did they bother?
Well, I'm not a big fan of the guys proposing the modern extended synthesis because pretty much everything that they propose that evolutionary biology isn't paying attention to, it is anyway. So yeah. The only people who I've seen you have on, and I've watched a fair few of your videos, who come and talk about things like evolutionary biologists are, one, not evolutionary biologists, and two, generally seen as quacks by evolutionary biology for a good reason. They get their facts wrong.
Well, they get their facts wrong or not is a matter of opinion at the end.
No, it's a matter of literature.
No, they are people who have read the literature. So I would say if they disagree with other people who have also read the literature or contributed to the literature, then it is a difference in opinion. So the question here returns to the same thing. At the end of the day, taking this attempt to extending things by function, okay, so that any glimpse of absorbing food will be called a digestive system, and any glimpse of fluid moving in a body will be called a circulatory system, and even a photosensitive protein with a photosensitive signal transporter will be called an eye, and anything that responds to sound waves will be called hearing, then hard luck. You have completely destroyed the meaning of novelty in this process.
Well, that's the point, though. What I'm saying is I don't think you have a definition for novelty that you could use. See, I have a definition of novelty that I think works pretty well, right? It's anytime any organism gains a biochemical, sensory, or anatomical trait that its ancestors lacked. That's it. I don't have to worry about whether or not it's in a system. I don't have to worry about the definition of system, which is the whole reason we're having trouble here, is because your definition requires a system. So, for instance, if an organism gains a new biochemical pathway that enables it to take advantage of a new food source or to incorporate some other bit of chemistry into its, say, use it for, like, I don't know, protein synthesis, right? Like, now it can break down, so for instance, plants take in nutrients from the soil, but they don't usually use them for energy. They use them to produce more complicated biomolecules in their structure, like proteins and, things like lipids and things like that, right? So they're not eating it directly, but they're absorbing it. A new biochemical pathway allowing something like that, so non-nutritive or non-calorific nutrition to come in and be utilized, anything like that. Or say you developed a new anatomical trait. So for instance, let's say you developed a connection between the mother's body and the yolk in an embryonic development, which is a reasonable first step towards a placenta, in which the mother can feed proteins and sugars through the yolk into the organism. That is a reasonable step on the way to a placenta. That would be a novel trait. Developing indents in the skin that can exude some kind of like secretion. Like mammals have they have a lot of pores in their skin, and they produce things like sweat and oils and whatever, right? Any indent in the skin that could do that, that's a novel trait. Any ability for a protein to react to a new stimulus, whether it be chemical, physical, radiative, that can then be interpreted by the organism, that's a new trait.
Oh, yeah.
So by definition has a big advantage in that you don't have to ask ahead of time whether or not something counts as a system and then rely on what seems like just subjective wishy-washy, well, I feel like it's a system now. Instead, you can say, okay, is something new happening? Yes, novelty. The problem, though, for you at least, is that under my definition of novelty, which is actually a practical one, the answer to the question, does evolution result in novelty, is obviously yes.
Okay, I can give you an answer that will become a very good common denominator here, okay? So what you're essentially saying is that I will call something novelty regardless how small or how big it is, okay? It's as big as a system or as small as, accepting a new molecule from the environment and doing something new with it.
As long as that trait is not present in the direct ancestors of the organism in question.
Yeah, understood.
Right.
So now we will then say that those changes are essentially, that those small changes are essentially what eventually leads to the bigger changes.
Nope,
correct?
I'm not saying that here. I might say that in a different discussion, but this is just a discussion about the bare fact or the bare question of novelty in evolution. Can evolution, through mutations and selection, as well as the other accepted evolutionary mechanisms like neutral variation, genetic drift, things like that. Can evolution, as it's currently understood, result in novelty without the intervention of intelligent agents? That's the only question I'm answering right now. So if you want to say, I don't believe that can go from Luca to a human, I'll say, great. I'm not here to argue it today. Today I'm here for a very specific question.
Okay. So let me respond to that in very simple terms. What you are really describing here is that should we believe that some mutations can be beneficial or not?
Nope, okay? That's not what I'm describing.
That's what you're describing. Something will now consume molecule number 2 instead of only molecule number 1.
Novelty could be disadvantageous. Just because it's new doesn't mean it's good.
Good, but it has become disadvantageous in general, but in a specific context, it was okay, so it was fixed. Okay,
well, sure, that might happen, but what I'm saying is evolution can result in novel functions regardless of their benefit to selection. Because remember, you can also have things fixed for reasons other than selection. So, for instance, the founder effect could simply result in some mutation fixing that results in a novel function, but that is not advantageous in the current environment. That's a possibility. I don't think it's terribly common, but just I can't rule it out. So no, this is not about beneficial mutations. This, at least not per se. I mean, obviously some of these novel functions are going to end up being beneficial in some circumstances. This is not about common ancestry, of universal common ancestry. This isn't about human origins. This is about one thing. Can novel functions, or novelty in terms of function or anatomy or the things I described earlier, arise as a result of evolutionary mechanisms, including selection and mutation?
The answer is no. Why?
Okay. But
the answer is no.
But
why?
We observe it happen.
The answer is no. Why? Because what
we observe it.
Okay, let him finish.
Sorry.
The answer is no, because what you are the place you are extending it to the extent of small things will always need that there is a backdrop of a precursor of some sort that it has changed. So you are at the end of the day looking at a mutative change.
Right, but evolution is always about changing what already exists.
Not at something that is coming that has no precursor. I have given an example in my discussion, very clear example. For example, that not only in the humans, but I have given an example with the humans, that like 8% of the human genome is new, has no homologs.
Okay.
And I can extend this to other. I specifically said 1,700 genes.
But I'm going to give an analogy and then explain it as to why I don't think that you've adequately addressed this. Okay. Now, you're an Islamic apologist, right?
I don't know what that means, but I defend
you're an advocate for Islam. You would like everyone to convert to Islam, and you advocate for its adoption and practice, correct?
Yeah, you can say so.
Okay. Suppose I said to you, I can't accept Islam because horses don't have enough toes.
Okay, I will tell you that Islam does not mandate that you believe that horses have enough toes.
I agree with you. It doesn't. It's a silly objection. Similarly, evolution not only doesn't require you to believe that things come without being modifications of previous things out of nowhere, but it largely precludes that possibility. So for the same reason that rejecting Islam because of the number of toes on horses is silly, it is also silly for you to use as an objection something that evolution doesn't actually predict.
Except, yeah, go ahead. Complete what you're saying.
Which is to say that things simply arise out of, I guess, non-existence.
Yeah. So I didn't say, and I had. There was actually a specific question about that. I think it was from the audience, or maybe from Dan. I can't remember. That was whether or not at all I accept that there is novelty, and I said I do accept. And then I gave the example of the 1,700 genes in the human genome that have no homologs. So the question is not whether there is novelty or not, and whether there are changes that were not in the previous generations or that cannot be traced to an ancestor or not. This is a fact. We have things that have arrived to the stage that were not there before. Small things, big things, complete systems, etc.
Okay,
complete
so
category
then the
of
answer to
creatures.
the question is yes.
This is not the question, Dapper, at all. This is not I'm not proposing this at all. I am saying that all the mechanisms that are put on the table by the theory of evolution, regardless which version you are looking at, and regardless which mechanism, will not provide a proper explanation for this to the extent that it fulfills its purpose. The purpose of this theory is to explain the origin of species. If you cannot explain novelty and you still claim that you can explain the origin of species, then your claim is false. You cannot explain the origin of species without explaining
We aren't talking about species right now.
Okay, but the question that this debate was predicated on, right? Because this is the aperture of the debate, so I consider this a continuation of that conversation, is, now I'm quoting here, can evolution create novel features? So you just said things that would lead me to believe that if that is the question, you would answer yes.
If that is the question, I will answer no, but this does not mean that I disagree that novel features have come to the stage. I am saying that being a fact that novel features have come to the stage throughout history, and being a fact that those changes are so big and abrupt and cannot be properly traced in a gradual gradient through its lineage, then the theory of evolution is not providing a proper explanation. Exactly like when we were discussing the example of the eye when we had that discussion with Jackson. And okay, the eye has different versions. The problem is they are not available in a linear manner on a lineage. And then you will invoke convergent evolution. And I said this in the debate. Now you say the eye has developed 50 times or 12 times or whatever number of times in parallel on convergent paths.
I wouldn't know the actual answer to that.
This does not then become an explanation, because then you will need to explain each path independently. This is the whole thing. So the whole thing is, does the theory of evolution explain the emergence of eyes or vision? No, it doesn't.
Okay, I agree. That is an interesting topic.
That was the question.
Right, that's a great topic. And if you wanted to have a debate with that topic, can evolution explain the evolution of the eye? I would be willing to host that debate. Yeah, however, I want to repeat.
Yeah.
The topic that you agreed to debate and that I agreed to moderate for was, can evolution create novel features? Not can it create the eye specifically, or can it create Ahmed's favorite list of things that he has trouble believing? Now, hold up.
Of course. The problem, Dapper, here is that this list does not stop at the eye, but it will go to each and every single system that is known in any mammal or any insect.
Right, but now we're back to systems. We don't have a go ahead, Ness.
You will not be able.
Can we perhaps focus on an example? Can we perhaps focus on an example that we have observed?
So
let's see what Ness has for us.
Yeah,
because Neslik hasn't said a whole lot, so I want to give him a few minutes of uninterrupted time to give his point.
This was an example mentioned during the debate, and it's like the sit plus phenotype, like in the Lenski experiment, where you have a mutation or specifically a duplication event. It created a novel regulatory module that allowed this E. coli to metabolize citrate under aerobic conditions. Well, by the definition that Dapper has provided, this would be a novelty. So, and it happened in a laboratory. So this is precisely the example of what a novelty would be. But of course, I don't think Ahmed would accept this as a novelty, but we don't know what his definition of novelty would be. So then we have to also, of course, go through that discussion.
I would like to ask you to explain the drawing. I don't want to explain it because it is your drawing. So just explain the drawing, and then I can tell you or not whether this is novelty.
So can you repeat again?
If you would explain what you have in the slide in front of us,
all right.
I can then tell you why this will or will not count as novelty from my perspective, and why in this case I will say that even if this is novelty or not, that it counts or does not count to the credit of the theory of evolution. Just start by the explanation. I will tell you my argument.
Oh, and, as you can see on the like, you can see my cursor, right, Dapper?
Yes, we can see your cursor.
But this is an image that the Wikipedia provides. It's accurate as far as I can tell. Like this on this figure right, it shows the expression level of the SIT-T gene. It's the citrate transporter gene, if I'm not mistaken. Basically, this protein right here that transports citrate into the cell and allowing it to be metabolized. Well, in the non-muted version, it doesn't get expressed, or at least not at a very high level under when oxygen is present. But in the mutant version, it is expressed, this gene, under oxygenic conditions. What happened? Well, basically, you see here the SIT-T gene, which under this SIT-T gene is under control of a promoter, which is silenced when oxygen is present. So, but then in the mutated version, you have a duplication event where the SIT-T gene comes, or at least a copy of the SIT-T gene becomes under control of another promoter, a copy of the RNK promoter. And in this novel module, as it says right here, this allows for the expression of this particular gene even when oxygen is present. So that is the novelty. It's not the protein itself is not the novelty. It's just that this regulatory module, a copy of the SIT-T gene being controlled by a different promoter, that is a novelty.
I think Ness is good, so I think you can go ahead, Ahmed.
Okay, so there were little problems in the sound, but I almost got the majority of what you said. So now let's look at this in the context of what we are looking at. So, and I actually have described my objection to this matter briefly in the debate. So this is E. coli, if you're referring to the Lenski experiment, okay? And yes, you are, right? And
yes, it's a long-term evolution experiment by Lenski.
Yeah,
right?
yeah. So you take the poor things and you put them in this deprived environment, and you there is an abundance of citrate, and you starve them from sugar, from glucose, I think, if it was, and then. And then, bam, those guys are metabolizing citrate. And you have rightfully said, just like I said in the debate, that this was not like the let's use this word. This was not an innovation of a digestive system for citrate. All the machinery was there. Cell knows how to digest, to metabolize citrate, except that the transporter protein is not enabled.
That is some of the machinery, so not all the machinery is there.
Yeah, so they are missing just to get citrate in, and this is what happened. So now let's fairly look at what was, and the drawing that you are showing on the screen was not known to Lenski from the beginning. So it was bam. E. coli is metabolizing citrate. Wow, here is evidence that under selective pressures, completely novel functions will come to the stage. And then after further investigation, no, it is not a complete novel function of doing everything that it takes to start from a citrate molecule.
That was never the claim, though. That was never the claim. It was never claimed that the ability to metabolize citrate in general is novel. It's just that this particular module right here, the regulatory module of the SIT-T gene under control of a different promoter, that is the novelty, which allowed for the expression of this gene under oxygen when oxygen is present. Normally, when oxygen is present, this wouldn't be allowed. But then, because of this novel regulatory module, now you can express this gene when oxygen is present. That is a novelty.
So now can you make the zoom out so we can see the drawing up there? Yeah. So at the end of the day, is this transporter protein an act of a Darwinian mechanism here? Is this random mutation and natural selection working on it? Or has this been classified even, I don't like the term, actually, I consider it one of those hollow terms, by Lenski himself as an acceptation?
Those aren't mutually exclusive things.
No, I'm asking you. I know those are mutually exclusive things. Which one was the classification eventually?
No, they're not mutually exclusive. An acceptation is an example of something working under the current model of evolutionary biology.
I'm not saying that acceptation is or is not under biology.
No. It's under evolution is what I'm saying.
No. The matter that anything that becomes an explanation to something is hijacked by the evolutionary discipline to be called under evolution is something completely different.
Right. What I'm saying to you is what you're saying is sort of like saying, is what you're talking about under the heading of the transmission of my car, or is it under the heading of automotive science?
No, the
acceptation is part of evolution.
The question, no, it is not. Acceptation is the function existed, and as a response to something, the cell activated the function.
Nope, that is not what acceptation is.
This is not something that happened by chance or by random mutation. There is nothing random about it at all.
That is not what acceptation is. Acceptation is when some trait is already primed to be used for some unrelated trait, and then a mutation occurs which allows it to do that thing. Now, that's not a very... Acceptation is one of those terms where, like, you can argue whether individual instances of acceptation really count. But an example might be when eccrine glands were co-opted by mammals for, in addition to protecting hair, being used to feed offspring, because mammary glands are in fact modified eccrine glands. Now, leaving aside whether or not you accept that story, if we assume it's true, that's an example of acceptation.
Yeah.
I'm referring now to this specific example, which is a very good example because it has plenty of empirical effort and plenty of empirical study. This is like 12 years now, or even 20 years. I don't know. I started when this one, 2003, 2010. So this is at least 12 years now. Okay. So at the end of the day, does this qualify as an event that occurs by chance? No.
Yes.
This is obviously a response.
Yes, it is. They have actually determined that because.
So are you telling me that the rest of the citrate metabolism cycle was there by chance too, sitting in the equilibrium, doing nothing for billions of years?
You're asking different questions. At one point you're saying, is this particular thing a result of unguided mutations? And then when the answer comes back yes, you're then asking a different thing, which is to say, well, is everything previous to it automatically just randomly there and it showed up? Look, dude, when we ask you about a particular event, do not bring it back to previous events. We can talk about previous events as a separate part of the conversation, right? But when you say let me finish, Ahmed.
Yeah, go ahead.
When you say, is this a result of a random mutation? What that means is going from the genome that's represented by the top to the genome represented at the bottom, the result of a mutation that was not directed by an intelligent agent. The answer, unless you're going to propose that God stuck his finger into one of these flasks in the Lenski long-term evolution experiment, the answer is yes. So unless you want to argue that this is a direct act of Allah, the answer to your question is yes. And you saying, okay, but I don't think that explains the top part, is irrelevant to that question. So now that you know that the answer to your question is in fact yes, unless you want to propose a direct miracle in Lenski's lab, now we can move on from that. So are you proposing a direct miracle in Lenski's lab?
I'm proposing that this is very clearly not anything unguided at all. I am proposing that this is a response by E. coli to a change in an environment, and this response is very clearly pre-programmed.
So if I take any collection of E. coli, there's a prediction.
It is decisively false. Yeah.
Right. Ahmed has made a hypothesis that we can make a prediction from. Okay. So if this is a response that E. coli undergo
just let me complete what I wanted to say, because you misinterpreted what I said when I asked about the rest of the machinery. Because
we're not getting to the rest of the machinery yet. We're still trying to figure out this part.
Because this I was using as evidence, okay? That this is not an unguided event. This is just the enablement of the starting piece of the process, which is the transport. And the evidence that this is not unguided is that, you have cars that work on automatic, but you have this button to change it into manual. And the matter that you press the button when you need to go on manual, it's not a surprise that the rest of the machinery that are needed for manually driven cars are there. Okay? You see where I'm going.
Okay, great. So now continuing with exactly what I was going to say, because all of that was a bit redundant. If this is in fact a response that E. coli undergo, is a result of being in a glucose-deficient and citrate-rich environment in the presence of oxygen, which is what happened in this particular line, then what we should expect is for this phenotype to be common in lines of E. coli raised in these environments for the length of time involved in getting to this point. But the problem is that we know that that isn't what happened. Only a very small subset of the lines in the Lenski long-term evolution experiment resulted in CIT plus bacteria. Other lines had other adaptations. So your statement resulted in an actual prediction, which is great. That's what we want in science. We want statements that we can test. The hypothesis, this is a response to the environment that is built into E. coli. Prediction, E. coli should typically undergo this change in a similar environment. We've already run the experiment. The answer is no. So this is not a programmed thing. This is not a response that E. coli just naturally do in this environment. This remains, unless you want to propose a miracle, because we've already done all the experiments, check. So short of a miracle, this is in fact an unguided mutation that resulted in this phenotype.
Yeah. We can clearly see that this mutation was specifically a tandem duplication event, because of course it's not very clearly shown here, but you can see here this CIT gene is a direct copy of this CIT gene, and what is located right, you can see it here, it was located right before this is a copy of this promoter right here. So it's basically, it copies this section from this section, and it sticks it right beside it, and that's why you also have a copy of this gene and this gene right here. So it's a duplication on this part.
So I actually, the response to what you have said about testing this hypothesis is not so difficult.
I agree.
Because we know very well that microorganisms, even when they are of the same species, are not identical. So if you put a bunch of bacteria in a flask and you apply an antibiotic, some of them will develop antibiotic response and some will not. The ones that have the correct plasmid will. The others will perish.
Wait, the correct plasmid?
So it's I'm sorry?
The correct plasmid? Do you mean the correct genes?
Yeah, the correct plasmid that has the correct gene that will deliver the antibacterial function. And we know that at a point, either other bacteria will have a plasmid exchange with that bacteria, or they will all perish, and the one that has the right gene is the one that is going to survive. But proposing now, so now we have
here the other plasmid. If I'm not mistaken, in the E. coli experiment, they removed all the plasmids, so it's not.
I'm not saying that this applies to the E. coli experiment. I'm telling you
that. Let's focus on this example.
No, I am actually focusing on the case. This is a microorganism. Our experience with microorganisms is that their response, even if it's the same bacterial species, that their response to changes in the environment, to stresses, are not the same. And even we see this with larger creatures, but it is very clear to us in microorganisms like E. coli, like bacteria, under different stress conditions, that they do not react the same and that their genetic capabilities are not the same.
Okay.
The overall genome of
that doesn't apply to this situation, because
it exactly applies, because now
guys, real quick, we're getting close to the closing time. So
yeah.
I'm gonna ask I'm gonna point out again that this is this is a hypothesis that Ahmed is using, which has predictive power, and we can check to see if the test has been done. It has, and I will describe it. So, the claim is that because there is diversity in any population of a single species of a given microorganism, presumably single-celled, in this case bacteria, right? Some of them will be predisposed to having this particular adaptation. The thing is that samples of the previous generations in the long-term evolutionary experiment are kept, and with it, when this feature was noticed, their previous cultures were revived, and it was not the case that they all could simply result in this. In fact, we found successive stages where it required mutations and genes that were sequenced actually occurred. And if you look at the ancestral populations, because we have generation 1 samples that are still preserved, you can still revive them and grow them, and they did. None of them can we find this in, nor can we find any of them which will be able to metabolize citrate. So the claim that this is just a result of pre-existing genetic diversity has already been falsified by the long-term evolution experiment before the hypothesis was proposed. So that explanation also fails in the face of the basic data.
I would like to give a 3-minute closing statement on this, because this is a very nice discussion. I actually wanted to discuss the Lenski experiment at length. You have two competing hypotheses. It's very simple. One of them is that this mutation happened completely by luck, completely unguided. It just happened that this blue part of the genome moved exactly in the right place, and then another promoter got copied and put exactly in the right place, and this amazingly, stunningly, produces exactly the right transporter protein that will complete the cycle of the already pre-existing machinery to complete the metabolism of citrate. This is one hypothesis. This is what you are advocating. Well and good. Somebody can believe it. It's okay. Now, the second hypothesis that I am putting in place, we have a very obvious gap of ignorance and misunderstanding and less understanding and no understanding of how microorganisms are behaving. Every day we know more. We cannot today, given our level of knowledge, draw a line at what exactly causes a specific set of E. coli or of any other bacteria to deliver this kind of response, and after how much time and under how much stress. We don't know. However, it is very clear to me, given the collection of evidence, that the rest of the cycle exists, that this happened, considering this a strike of thunder or of luck seems very remote to me, that all the constituents are there. This is essentially, for me, as somebody who works with software that is intelligent enough to rewrite its software to deliver the right response in the right time. Now, the only question is, why this right time is 1 year, not 1 month, or not 1 day, or not immediate? Why does it happen in 2 flasks or 2 containers out of 12, or 5 containers out of 12, or whatever? Why did it happen in that specific generation in flask number 5 and that other specific generation in flask number 7 are gaps that we can cover in our ignorance, instead of accepting the other hypothesis that this whole thing is by chance. This is the whole thing. This is, yeah, sure. Yeah.
All right.
Wow. This is a streak. You will win a streak this way.
Yeah, I'm glad that we got to discuss this topic too. Like, I think it's now clear that we both agree that this part right here that is highlighted is a novelty, right? Because you have a different a gene under control of a different promoter. But of course, Ahmed is
hold on, there was no
with...
audio.
Oh,
I'm an idiot.
Oh.
Every once in a while I mute to cough and then I forgot. So for $5 from my unfinished journey, he says, since, quote, nothing in biology makes sense except in light of evolution, to what extent is the term evolutionary biology redundant? And I said, it's a little bit redundant because, like you said, everything involves evolution. However, if your thing is like, oh, I'm going to study the protein folding problem, or like I'm going to be an expert in the anatomy of the green iguana, then it's not very important that you know lots and lots about evolutionary biology. You should know some, but you don't need to be an expert in it. And also, knowing everything about, say, how mutations and selection and neutral variation and drift and all these things work doesn't itself get you, say, the solution to the protein folding problem or, like, to the detailed anatomy of the green iguana. So, there you go. Sorry, Nestle, go ahead.
No, I was just saying that I was also glad that we can discuss this topic. Like it seems to be that we both agree that this particular region, like highlighted here, is a novelty because of you have a gene under control of a different promoter. But Ahmed is disputing that whether this mutation, this tandem duplication, is actually for lack of a better term, random or a directed result of something pre-programmed. Well, but the Lenski experiment, like they kept, as Dapper mentioned before, they kept frozen samples of the ancestors, and they examined them, whether they would have the same response. And well, the further back you go in the ancestry, the less likely they will have the same mutation. So it seems to be that this mutation is very dependent on undirected mutations. So, but of course, Ahmed seems to be adamant into refusing to believe that this can be undirected. But that to me, that seems to be an argument from personal incredulity. He is personally unable to see how this can be the result of an undirected process. And that's my final thought on this.
All right. Well, in that case, we are going to go into some channel announcements. First, I want to say that as one of the debaters from the debate, Ahmed's channel is, in fact, linked in the description. So I encourage everyone to go check it out. I watch it, so why aren't you? Let's see. Also, Creation Myth's channel is, in fact, linked because he was the other debater who didn't make it here, but that's okay. So please check that out. Again, I watch that channel, so why aren't you? Also, yes, F's in the chat for my inability to actually unmute when I'm supposed to, because sometimes I mute and have a little cough or clear my throat, and then I just never unmute. Because instead of like a cough button, I just have a toggle on OBS. So yeah. Hey, you know what? Plug for my my Patreon and channel memberships. If enough people join that, I'll be able to afford a dedicated producer. So there you go. Let's see. Ahmed, do you have anything coming up on your channel that people should know about?
Yeah, so I'm not only doing evolution these days. I'm jumping into philosophy because it relates to religion. So I have had a couple of interviews with Pat Flynn, who is a U.S. Philosopher, and I also had a remarkable one with Dr. William Lane Craig, and we discussed the Kalam cosmological argument. And coming up on 3rd of November, we will discuss divine aseity, which is a pretty nice topic. On evolution, I had Dr. James Tour, I had Dr. Faz Rana, and later I will be having Dr. Gunther Beckley, who is a paleontologist, discussing the fossil record. But this is coming like mid-January. It's a bit far from now. And for those who would like to listen to a structured argument for why evolution is not exactly correct, I'm having this structured step-by-step course that I call Is Evolution True or False? It's fun, and I do a live stream almost every other Saturday, or actually 1 week after the session. I considered our debate today to be a session, So
Okay.
I already announced right after it that I'm doing a Saturday stream this Saturday for people who would like to either subscribe to the course or come at me with questions or challenges about novelty. Please do. You're welcome. The course is free and everything that I do is free. So this is just about it, apart from a series called Knowing Islam that I explain Islam in very simple terms.
All right. Nessie, you got anything coming up?
The most work I do online is helping Jackson read with his videos. Although lately, I have also been busy with my education, and Jackson has been busy, so we haven't been able to put out a lot of videos lately. But Jackson says it will change in the future, so stay tuned.
Well, tell Jackson that I currently have a signature from R.J. Downard on my copy of The Rocks Were There, but I'm still waiting for my Jackson Wheat signature, because they're both authors on that book. So, with that, I'm just going to give a quick update for what's coming up on my channel. So, let's see. Actually, not on my channel. After this, I'm going to be doing Guns, Magic, and Steel. That is my D&D game, where we stream it to Cheshire Vic's channel. Maddie from Science Side Up is in it. Erica from Gutsick Gibbon is in it. TD Lane, who's appeared on this channel, is in it. Bent Hovind, who's my frequent co-host, is in it. I'm in it. Gutsick Gibbon's fiance Luke is in it. So go watch that. It's going to happen in just under an hour at 4 p.m. Pacific, which is like, what, 7? Yeah, 7 Eastern. I don't know what time it is in, say, like, I don't know, Egypt or something, but whatever. So go check that out. Tomorrow, we are going to be doing a Matt with Maddie. So that's Matt Powell videos, and we're going to be taking a look at it with Science Side Up, Maddie. Thursday will be the next Speculative Evolution video, so if you've been waiting for that, well, you're in luck. Saturday is going to be a Leaving Young Earth Creationism. And then, let's see. Then next Tuesday will be a Kent with Bent. The Tuesday after that, will be a Kent with Bent. Then there will be the first video of the Helping Cindy Lincoln series out. And Cindy has agreed to watch it. So that's good, because it is directed at her. So she is going to be watching it. And also, she and I are talking about having her on sometime for some kind of discussion about a scientific topic where she and I disagree. And so, which reminds me, I interviewed her about the abuse that she suffered at Dinosaur Adventureland at the hands of Kent Hovind, her then husband. And it was an extremely good interview, but it's heavy. If you haven't seen it yet, I highly suggest that you do it. But recognize that it's an upsetting interview. You're not going to come away from it happy. But I still think it's important. I don't have any word from her as to whether or not she has or is likely to in the near future set up a GoFundMe for legal fees. That's up in the air. Or even a GoFundMe for things like getting her out of within walking distance of Dinosaur Adventureland, where she has a protection order out against the leader of that place, but he could literally just walk to her house. So, I don't know. That's still up in the air, but please do go watch that. I think it's one of the more important videos I've put out because things like this are trivial compared to, a woman ending up with thousands of dollars in hospital bills because her husband shoved her to the ground. And a cult that's taking advantage of people's wealth and then spitting them out the door the minute that they question the leader, where now they're destitute and have to beg to even be able to survive. So, yeah, those are important things. This is trivial compared to that. But with that, I'm going to leave you guys. Thank you very much once again, Nessie20 and Ahmed. And Dan, I want to say thank you, even though you didn't make it. All right, bye, everybody. I just want to take a second to thank my channel members and patrons, especially those pledging $20 and above. Bob Knob, Bent Hovind, Ian Chen, Chris Love, Sphincter of Doom, The Venerable Bede, and Res Instance. My channel members and patrons help make this channel possible, and without their support, this channel really wouldn't exist. If you would like to help support the channel as a member or patron, there are links in the description to both join the channel as well as join the Patreon. Patrons and members get mentioned in these credit scenes, as well as getting early access to my scripted videos, and if you pledge $10 or above, you can also get access to various 3D assets that I create for Blender, both for use in the channel, as well as just general giveaways to my supporters to help in any Blender projects they might have. If a monthly or annual pledge isn't for you, then there is also a merch store linked to the description, and if none of that's right for you, please just like and subscribe, because every like and subscription really helps the video out. Thank you.